Hello guys, my name is Minhajul Islam from Group 324 with a topic translation from the book Walkenstein. Translation, which is the conversion of polynucleotide transcript of DNA and mRNA into the amino acid. Protein text occurs in the complex of ribosome with a polynucleotide chain that is in polysome. Ribosomes of eukaryotic are characterized by a high value of 80s and not 70s with a molecular weight of 4.1 to 4.7 into 10 to the power 8. The 70s ribosome consists of two subunits, 30s and 50s, where ribosomal RNA constitutes about 65% of the triweight of ribosomes, the remaining 35% being proteins. These RNA are subdivided into three classes. Presumably, the protein molecules interact with the non-helical portion of RNA, and the ribonucleotide protein complex is coiled into the compact structure of the ribosomal subunit. The 70th ribosome contains with a 65 polypeptide. The complete assembly of ribosome and their reconstitution from the proteins and RNA has been accomplished by NOMRA. The role of RNA is specific but not absolute. The functional 30S particles are produced from 16S rRNA, one of the species of bacteria, and ribosomal 30S protein of another species. The number of proteins as isolated was 19. The absence of any of these affects the function of 30S particle. All the protein function is in a concerted manner, which is also necessary for self-assembly. The kinetics of self-assembly corresponds to a first-order reaction, which implies the presence of certain slow intermediate step which limits the reconstitution process. The strong dependence of the rate of self-assembly on temperature shows that the reconstitution to the effect of a free energy of about 160 kilojoule per mole of ribosome is needed. The self-assembly of ribosome from all the component in vitro takes about 5 minutes. Translation begins with initiation, that is the synthesis of first peptide linkage. Two amino acid tRNA complex are stimulated involved in the initiation. This process repeats itself many times on each ribosome. Cyprian proposed a spectacular module of the operation of the ribosome. It is assumed in this model that the amino acid tRNA binding site and the peptidyl tRNA binding site are localized at different subunits at 30s and 50s respectively. The peptide transferase center, which provides the transfer of peptide, resides at the 50s particle. The ribosome is periodically opened and closed. A mathematical model that describes these processes allows one or to calculate the distribution of the size of mRNA in agreement with the experiment. On the whole, the protein biosynthesis is determined by the rate of transcription and translation. Thank you.